Okay, so welcome to week 5, preliminary data, all about it. So we're gonna have a little quick chat about that. So you guys have done your connection with each other, which is proposal. This is kind of actually my main slide I wanted to show you all. And these are different components of the proposal that you're writing. And so if you look at it from this perspective, it presents itself, it presents units that you can closely analyze. And I think it is that I would like to suggest to you that these are areas where you can perhaps extract and begin to ask fundamental questions about, is this making sense? Is this clear, right? What is the potential for this to actually have the broader impact on the intellectual marriage? And the cool thing about this is that the actual NSF proposal, the reviewers, right, they go through this in the same fashion more or less. And they rank them and they give you this scoring about, is this fundable or not, and so forth. So, of course, we're not going to do that for you guys, but as you're building a proposal, I think it's useful to think about who the audience is, as we've been talking a lot about, because those are the people that you should reach out to, even in email or some, if you have a cluster of them, even better, you can send them a survey, very brief, few questions, asking them, hey, can you take a look at this outline of the proposal and give me feedback? And in fact, if you can get some written feedback, right, that is itself preliminary data. It tells you, are you going in the right direction, right? Because right now you've had your coach, yes, but try to venture outside and get some insights. I mean, maybe from your informational interview, actually. That's a little tip there if you're pitching them. Say, hey, would you mind reading this thing and give me some feedback on what you think? But I say go after your key audience, about who that audience is, and try to extract them. I'm using this because Abigail and I've been talking a lot about this. We're talking about our audience, and so it's really useful to think about that. And Abigail, if you think the paper you were just reading, you and I were reading, from users for others, we should post it on the channel if I were to read, because there's a really nice way to visualize this component. So, transitory science and all the different components. So anyway, so the background. Background is an element you can extract, right? Is it clear? Does it set the stage for understanding the bigger scope, right? So that's your audience can give you data on that. The research questions themselves, are they specific? Are they targeted? They can lead to fundamental better analysis of more knowledge in particular area. The design, and I think this is where you probably you want the most information is the design aspect. Does it make sense given the questions? And hopefully over time, so far, you've been iterating that and making it better. Or is there a better way to approach it? And so that's something you can really get data on. Right? Also, the different tools you could use, focus tools, qualitative things you can do network analysis, many ways. But ultimately, it has to be able to address the question that you are trying to address. Then the intellectual merit. Now this gets into, well, is this going to help us get new information, new knowledge, right? Something innovative in your approach, right? And so really the audiences can give you insight on whether that's the truth or not. Same thing with product impacts. And I would say the intellectual merit, probably the people perhaps are in the field already, people you're reading, the papers you're reading from, right? So you can even think of Katie Canfield, who came to us. I think she'd be a great one if some of you email her and say, hey, here's a preliminary thing. Could you take a look and let me know what you think of the following components, right? You don't have to do analysis of all of them, but you can pick and choose which ones you want feedback from, right? And then that is information that then in your pitch, in your proposal day, you can say like, hey, this proposal actually, we ran it by a few people. Here's what they're saying. They're saying actually, yeah, this design may give us better insight, blah, 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 or they gave you advice and you change course along the way because they told you X, right? That is information that you can use in designing a study. And this is actually part of the normal process. If you're writing proposals, right, Jess and I have done this many times, you would take it to some people for feedback before you submit it to a grantor. So you want to do that. You don't want to wait till the last minute. You want to get it in early, get some initial feedback, and then iterate and change. Okay. So I want to give you some. So in your resource folder, some of you may have noticed, I've added a sample bank of questions you could use. All right. So I know this could take, you could think, oh my God, I have to design a whole survey. No, you don't do that. I have sort of made it easy for you. So I want to show you that right now. Okay. This is something you can use just to give you a framework of analyzing different components. So let's say you go ahead and you either talk to someone, an audience member, an audience of your potential proposal, or you identify a group of people, you have the emails, you write a quick summary, and you tell them, listen, I'm writing this proposal, we love your feedback, and here's a link to provide your insights. And you can imagine them coming in, their name, email, fine. And then overall, how would you rate the framework, creativity of the proposal? These are actual, some of these are taken from the NSF, the analysis framework. How would you rate the background of the proposal, what's missing in our understanding, like someone telling you that. So you require an expert to really get in here to understand that. But if you're pitching, depending on the audience, it really depends on who the audience is. And all of these are different audience levels here, so keep that in mind. Clarity of the research questions is a huge one. It doesn't make sense. Sometimes it doesn't. The intellectual merit, so it actually explains encompassing the potential to advance knowledge, and what do they think about the potential of the data, to move us one way or another. And all of these are just preliminary insights against the proposal. Hope you guys are understanding that, right? It's not preliminary data on your actual proposal, which if you can get that, sure. And I'm very curious if any of you are thinking you might be able to get any preliminary data on your actual proposal. I'll be very interested to know that. Because if you do, absolutely share that, okay? But I'm willing to bet most of you will not. That's an additional bonus. But please tell me if that's true. The broader impact, same thing. And this is just a quick sample. You can take any piece, you can have a conversation with someone, for example, and then ask them just, hey, in my pitch to you, what made sense, what didn't make sense, right? Get that qualitative data, right? But from here, you can get some quantitative assessments very easily on the different segments of the proposal, and get that initial feedback that you can share on your pitch. So, questions so far. I've been trickling this throughout the week so far about the preliminary data, and today is really the formal thing where I'm telling you the analysis, the meta-analysis of the proposal is what should, I think, explore and getting some preliminary data for your presentation. Does anybody have any questions, or if anything does not make sense, please speak up now.
So should we be trying to collect preliminary data on, we mentioned in our meeting, sort of just presenting an abstract of our proposal, or should we present like the entire proposal?
Yeah, so you guys are in different stages. So depending where you are and what you're comfortable sharing, that's what I would suggest you do. If you have an outline, again, from what we discussing, for example, in your Google sheet, I feel like that's enough already. You have some ideas, high level ideas, right? I feel like most of it would be a conversation if you can have with people, teach them as you take them through it, and then get them to answer this after the fact. Or if you feel like, my God, I don't have much, you can target one area and then tell them, can you give me feedback on this? Here, most of them we want to get the whole framework I shared earlier, right? Yeah, so hopefully that's cool. Is anybody thinking of having actual data from their proposals themselves? Okay. I feel like Daniel, maybe you probably be the closest one. Maybe.
Yeah, I was about to say I'm thinking about something I am collecting is sort of like just general statistics on TikTok usage, and then also like likes, comments, interaction, shares on certain videos. So I guess that qualifies as preliminary data for sure. But I think also something I'm thinking about is surveying TikTok users to understand, what they've, the information they've gotten from TikTok about COVID and things like that.
Yeah. So existing, as we discussed this before, I think week 3 or 2 or 3, existing databases that have that information for analysis, right? Abigail, you and I were chatting that there is some, I think for you, there's qualitative data somewhere, right? People's houses or written text that you can examine. And that itself is something that you can showcase as well. Data that's partially there and say it's partially there, but there's a whole world that's missing, right? As well. Chloe, what are you thinking?
So I'm also considering doing a survey, like maybe just like broader level questions of what I would potentially ask about in my actual proposal. So sending that out hopefully to schools that have implemented a school garden curriculum versus ones that haven't. Just like a very short kind of overview of what would actually potentially be asked.
And then you want feedback
Yeah.
for that. Okay, so
Yeah.
recommend you try to get that like sooner versus later.
Yeah, I'm trying, but I think a lot of schools just went on summer break, so I'm not getting responses, but I will continue to try to do that. If not, I will figure something out.
Reading about preliminary, the ends are kind of small.
Yeah.
You don't look for significance here.
Yeah.
It's really about trying to figure out you're playing in the right direction, For your proposal, number one, that's one dimension, right? And then proposal design or the actual proposal itself for the actual question that you're asking. Two different things. Okay, try to separate them slightly here. Both as well. But yeah, Jacob, thoughts?
Yeah, in terms of, I guess, preliminary data, I'm still kind of potentially, I guess, figuring out like, because I kind of know my target audience is kind of going to be like other educators in the field of children's nutrition ed. So like I've done a couple informational interviews with people in that area. So maybe potentially I could kind of send out some, I guess, like the core ideas of my proposal to kind of get feedback from them. In terms of, I guess, I'm not sure if I'm going to really be able to get data like that will support, I guess, like the questions, like the experimental data. I'm not sure I'm going to be able to get much from there.
And that's fine, as long as you know. I was still looking to existing data sets within the questions, or you're asking diverse sort of questions. And it's not for every single question, maybe for one of them, right? That may be able to get some insight. And if it's missing, it's missing. Abigail, yeah, I feel like we just talked about it recently, but not everything's there. So you want to share your high-level thoughts?
Yeah, so I'm also planning to sort of pitch maybe 10 to 20 people who are familiar with the field that I'm working in, who are familiar with the co-production of knowledge, pitch them my proposal and try to get some survey responses back, just generally about what people think about the research questions and approach and everything. But yeah, there's definitely limited data to collect right now, because part of my question is like, we need more data, pretty much.
There's lack of data, right? That's kind of your you're going in there with that. But that's good. This is all great. And if there's something that's missing, something that you may come across, I think just feel free to include it. Diversity of data sets. But in this field, there's multiple ways you can look at something. So now I pretend you have this data, right? You're now trying to put it together, right, into a narrative, right? Whether actually in your proposals, you can put some figures in there, right? Or in your presentation, with different figures. Turns out that's actually not an easy thing to do. Right? You can take courses and so forth, but the best way is to push practice. Reimagining figures, reframing them, asking yourself, what am I trying to say? And we have an expert at this. Jessica, I don't see her papers. I'll just encourage you to Google her name and see her papers. Just amazing, because the figures are very complex, but they're easily understood and easy for you to present it. Very like Apple pencil like. So with that, I wanted Jessica to just give us a sense of how she thinks about figures. We're not going to teach you how to use Illustrator. I think that's not going to be the point here. It's really about how you think about figures and approaching them. And if you don't know how to do something and how what tools are available, we'll have a discussion about that. So, Jessica, if I didn't miss something about your introduction, please feel free to say a few things. So just
No, that's perfect. I think the goal of this, it's like a short presentation with some slides, mostly so you can see what some of these figures look like. And the way that I sort of like to think about figures and visualizations is that one, you're trying to portray your data accurately, right? So you need to do that in a way that shows the point that you're trying to make. But two is also to make it so that the reader, or if you're giving a presentation, isn't sort of like appalled by your figures, right? So I'm sure you've all seen presentations where you're like, wow, that's really an ugly figure, or why did they make it like that? Or it's just like very jarring on the eyes. So you want to be able to convey your own data, and you also want the reader or the audience to not be mad at you, basically. And a lot of the slides I'm going to show you are based on some kind of cool design principles that someone at the Broad Institute in Cambridge is like basically a designer by training who has taught a lot of scientists there how to make very visually appealing figures. And so if you want more resources, he has like tons and tons of different papers that he's published, and all of them are just on these like basic design principles. So I'm going to share some of them with you today, but you guys should feel free to pretty much interrupt whenever, or if you have questions, or you have an idea or something like that. Okay. So the first sort of visualization principle is this concept of the overview figure. And yeah, you know, and a lot of these are very like basic science focused, but you can really apply them to any type of research you may be doing. So the idea of the overview figure is you're trying to give the reader or the audience a sense of like the overall arc of a method or a project. So for many of you, it may be, you know, this is how the research proposal overall arc might look, or this is how the survey arc might look, or a specific program if you're going to recruit people for it. It's just to give a sense of a broad overview. And I think this is a nice example from this paper, because you clearly, most people read from left to right, at least in the United States. So you can see kind of left to right where you need to go. And the colors are such that they're not like hurting your eyes. And it's very clear, I think, what it wants you to see, right? So each step is labeled with some words, but it's not too complicated. And each one is like a single step. So I think Bang Wong is this guy at the Broad who puts together all of these data visualization principles. And he sort of feels like this is a very nice schematic overview of a fairly complicated method that these people are trying to convey in a scientific paper. So sometimes thinking about how to make that overview is actually more difficult than you may think. Another example of an overview figure can look like this. So this is for vertical. So it's going top to bottom. And this is also describing some kind of a method. It looks like some, you know, shRNA plasmid library that they're doing some kind of screening for. But the nice thing I like about this is that, again, it clearly flows in the correct direction. There's arrows indicating which way you should be reading, but the colors also match and make sense, right? So they're all here as you move down the schematic. And then there's words to prompt people to read. So what you'll also find is that there are some people who are more visual learners, where they actually, if a slide goes on a screen, they start reading immediately. So I'm one of those people. So if you put a bunch of slides up and all the text is there, I will stop listening and I'll just start reading. There are other people who don't read anything on the slide and they only listen to what you say, right? So you kind of need to be able to cater to both of those types of audiences. And in some ways, figures like this do, because if I'm just going to read the slide when you put it up, I can actually understand everything that happens. But if I don't read any of it, I can actually listen to you as you go through each of these sort of pictures or schematics. So I think thinking about big overview figures is more difficult than it sounds, but is important. And for a lot of people's papers, this is either the first figure or the last figure, right? They're like, oh, we developed this model, or this is how we think this works, or this is a summary of all the data that I collected, or the process by which I went through. So more specific design principles. So layout is actually more complicated than you would think. So if you look up the definition of layout, it really just means arranging text and images on a page or on a slide according to some aesthetic scheme, and the purpose is to like very clearly clarify your presentation. So like in the graphical art world, the layout is trying to point towards the elephant in the room, right? This is like the point that I'm trying to make. And so when you're trying to communicate your research or your project, actually having a good layout is super, super important to guiding readers or the audience into some complex material. And this is stuff that you probably have already seen before, but is a little bit more explicit, which is this concept of the golden ratio. So the golden ratio is this 13:8 ratio, and you can see it sort of outlined here, but is used in many, many things, including art. And so you can see that it's used like as a very visually appealing ratio that as humans, we use visual, like this 13:8 ratio is very, very aesthetically pleasing to us. And so even thinking about things like this in making your figures is actually super, super important, because it just makes it easier for the reader to understand what you're trying to show them. There are other things about layout. So for example, I'm sure some of you might have made like poster presentations or in a PowerPoint slide. There's sort of practical applications of the golden ratio, even to these things, where you can sort of set up your slide or your poster as sort of a golden ratio, right? And these are just sort of schematics of ways that people have used these ratios to their advantage, like putting these figures in this particular format, or even ways of sort of guiding the reader's eyes. These lines are sort of where they want the reader's eyes to go to, and leading them through whatever story you may be trying to tell them. So often these grids can be good for layout and can help people follow where you want them to go. I like this bold lettering in all caps, the kind sometimes used to demand our attention in signs around the lab, read first, right? So you kind of actually want the read first to be in the beginning. So again, there's like ways of laying out your figures and your schematics to make it actually anchor where you want people's eyes to go. A lot of these are also like little neuroscience visual attention tricks that you can use to your advantage, which I'll show you in a little bit. So one of the super important principles is this sort of Gestalt principle, which is actually this like German psychology principle that Germans came up with in the 1920s that was kind of used to explain how humans organize visual information. So figures are all about visual representations, right, of your data. And it may be that you have a table, but you can still make your table organized in a way that's visually appealing. But these are more kind of shapes and figures and things like that. So Gestalt is actually a German word, and it means shape or form. And the principles that describe the way we visually assemble individual objects into these groups or wholes. So I kind of like some of these because they they show very nicely what we mean by Gestalt. So for example, in A, this is this idea of subjective contour, right? There's not actually an edge on this white triangle, but based on where the circles are, or these Pac-Man, whatever you want to call them, you can see the white triangle start emerge, right? So even without drawing the triangle. So that's kind of a cool effect. There's other ways to where similar objects are sort of visually grouped. So while these are the same size, most people tend to see the circles grouped together and the squares grouped together, or they may see certain sizes grouped together. So also thinking about that. And then objects that are placed closer to one another are often seen as going together. That seems like really obvious, but I feel like people don't say that explicitly. So these are just examples of the squares, and when you put them closer together, your eye seems to think that they're more related, right? So you can think about that when you're trying to put figures together, like even these little differences in space between the squares make them seem more related. And then the other thing to think about is like vertical and horizontal correlations with objects. So these are all the same little tiny black squares, but they're oriented in different directions and they actually kind of look different. And so you can sometimes play with orientation in your figures and just see how they look and if they look different to you or other people. There are other things about these Gestalt principles in terms of grouping. So for example, in 2A, there's this idea of sort of perceptual grouping where, so you have square, circle, circle, square. When I look at that, I'm like, okay, those don't look really related. But when you add the lines, suddenly they're related to each other. And then when you add the rectangle, my eye and my sort of visual cortex reads that as, the square is going to this circle, and this is actually the end point because there's a square around it, right? So there's ways that you can very simply sort of force people's eyes and vision into a certain direction to make your point. So I also like this example. These are all dots. These are all data points for some graph, right? Hard for me to really make sense of what they mean. I think that maybe the squares are probably supposed to be all related to each other in the circles, but they're not connected. So in the same example, the squares are all connected now, and the circles are all connected. And now I've added this red circle that indicates what I want you to look at, that whatever, they meet the same peak, or at this particular time is something big that happened, right? But these are actually the same figure. You've just added a couple things that make it more easy to follow and makes your point, which is whatever's in this red circle, right? So these are really, really, really simple design principles that actually can be really useful for drawing people's attention. I think this is another really good Gestalt principle, which is this concept of visual completion and continuity, which we sort of talked about with the triangle earlier, right? So this is like a very famous triangle that is called the Canizza triangle. I don't know if I'm saying that right, but which is basically an illusion, right? You haven't actually drawn the triangle. You can just see it based on the contour of these Pac-Man shapes that form these well-defined edges of a triangle, right? And then another example is here. So we automatically sort of spontaneously perceive that there's a full circle behind the square in this image here. But you don't really know that there's a full circle behind that square, right? So it could be that it's really just a Pac-Man. It could be that it's actually a teardrop shape, or it could be this like weird e-spudding thing, right? But for some reason your brain is like, oh, that's gonna be a complete circle. But it could actually be any of those things. So I think thinking about like how like edges work together and how we perceive these shapes to be continuous with each other is also like something kind of fun and interesting to think about. The other really, really important thing is alignment. And I see this a lot in figures that I don't like and in presentations that I don't like, which is basically that someone is just like putting random things on the screen and they don't align, and it really bothers my sort of like visual cortex and the way that people's brains tend to work. in terms of looking at visual material. So, you know, Bang Wong at the Broad often gives this advice to people, which is you should actually maybe even put up grid lines when you're making figures and make sure that they align appropriately. Or I think even in PowerPoint, there's a way to make figures snap and frame with each other. But actually show yourself and prove to yourself that your figures are lining up so that they're more visually appealing for people, right? So these are all different ways of alignment. So this is like vertical and horizontal alignment for this figure, but there's also curvilinear alignment that can really drive people crazy if they're not aligned on a curve. And there are many, many ways to use guides, whether it be in PowerPoint or Illustrator or Photoshop, whatever you're using to make, or if you're drawing a figure, there are ways to use these rulers to make sure that that piece is aligned. And I think it just makes the figures much cleaner when you put them together. The other thing people don't think about is negative space. So not just the actual figure that you have, but what about all the blank spaces around it? So this is just an example of some poster that someone at the Broad and Dana-Farber presented. So this is all the text and their pictures, but this is to try to give you a sense of exactly how this poster was planned out, like in the background, and trying to think about where this should all go and what all the negative space means, right? So negative space can direct your attention that, oh, maybe I'm supposed to read to the next column, or maybe I'm supposed to move to the right or to the left. So kind of using negative space to your advantage is also really important. And I think also shown here, right, is that, yeah, this is a figure of some like protein structure, but it actually also looks really interesting if you color it differently. And our eyes tend to go towards that white with the contrasting black. So just thinking about not just what you put on your figure, but what's like not in the figure, and that looks okay to your eye, and showing it to other people too is also can be useful. Things that might be more relevant to your projects for the internship are this idea of matching salience to relevance. So often I'll see figures where, like, you know, someone has a control condition and a bunch of experiments, and the control condition is like bright red, right? And then all the experiment stuff is like black or some more neutral color. And I'm like, why is your control like the bright color? Like, you should show me that your like really cool result is the bright color, right? So this is that point I was trying to make in the beginning, and that one of the goals of making figures is to convey your data accurately and make the point that you're trying to make. So this is just trying to say that there's a lot of different colors that have different like red, green, blue patterns, but your eyes immediately, this is a very simple example where it's highlighted sky blue, so your eyes immediately go to that, right? So I don't even really read black or orange. I immediately go to sky blue as my attention. So that's whatever this person made this figure wants my eyes to go to that. Or things like coloring particular aspects of your figure. My eyes are immediately going to jump to the red dots, right? That's the point you're trying to make. So in some ways, it's like putting all of your data together and making the figure. So someone maybe put all the dots together and made the figure. And then they looked at it and they're like, well, the point I'm trying to make is that the dots that are on the upper left are like the most important. How can I kind of draw your attention even more towards those? And that can be through color. They've shown this like dotted line that delineates some kind of border, right? There's lots of ways to emphasize the point that you're trying to make. But I think being intentional about matching salience to relevance can be very, very helpful. This is another very good example, I think, of matching salience to relevance. So you can see that something that's like, so this is the scale, and the scale shows that things that are low are like black, blue, yellow, and things that are higher, this dark red. So the point that Bang makes in this paper is that that actually doesn't make sense as a scale. Like the brightest thing on this page is like in the orange, like kind of yellow section. And that's like the middle range of this scale, right? So this is a situation in which the salience is not matching because the brightest colors actually don't indicate what you're trying to convey. So it may have been better if actually the brightest color was over here on the high side, right? So there are ways to change your scale to make your point. He's just saying that red and yellow and blue are kind of like primary colors, but. And then there's other things just about like visual style. So these two, A and B, are showing the exact same thing, but they look really, really different. Do you guys have like comments about how these look or ideas about how these look? Do you have a preference for one or the other?
All for A, raise your hand. I'll say, I don't know, I like A. Why? Yeah. I'll give my A vote. Anybody following me? No? Oh, man. Okay, B. I guess everyone's B then.
Why do
Okay.
you guys like B?
I like that it's like consistent throughout because there's a lot of distracting colors, shapes, and
Yeah.
textures in A.
Totally. Anyone else?
What I like about B is that for me, the emphasis is on the genes like A and D, which I assume are significant in this research, and then also cancer. So I think like perhaps the main point that the authors are trying to make about their data has been highlighted in that image, whereas in A, it's sort of all over the place.
Totally. I think the point that Martin is someone who also writes a lot in some of the nature journals about data visualizations, but I think the point that he was trying to make with this was that like, in some ways, each of these images standing alone would look great, right? But then when you put them into a figure panel, you're like, cancer is like exploding. And you know, you made the good point that like, why there's stars here and the colors don't really match. And this figure is horrible. I don't know why there's like Tetris blocks here, right? So in some ways, people always feel like, wow, I need to make my figure really fancy and really bright colors, and I need to make it pop and like look three-dimensional and things like that. But in some ways, like more simple figures can actually get the point across better and also be truer to your data, right? And not distract people from the point that you're trying to make. So, you know, I think there's also things that you could critique about this figure, but I think he was just trying to contrast those two, which is great. So this is another example of that kind of contrast between the two. So, you know, there's this panel here and then this panel here. And so one is that Venn diagrams can be useful to convey information, and you can do kind of, there's many ways of doing Venn diagrams in different colors, and you can kind of decide what you think about these. But I think the other thing is that there are ways to highlight similarities. So this is basically showing 4 different genes in this like Fox gene family, and this is showing the same genes. Do you guys have a sense of like what you like better? I don't really think there's a right answer to this, but.
I think if they're trying to say that FOS L1 is sort of separate in structure or some other element than the other 3, I think the one on the right makes more sense to me because it's shaded differently.
Totally. I think the point they're trying to make here is that a lot of times people like may label certain things in different shapes or different colors, and it actually totally detracts from the point you're trying to make, right? It's like, well, maybe Foxer 2 is like important because it's orange, but then this doesn't have a square around it, right? It leaves too much up to the reader. Like in some ways you want to sort of tell the reader, like, look, they're all the same. And like you said, Gestalt 1 is the one that I want you to look at, right? Again, this is a more simple version, but it probably gets the point across a lot better. And then this is another kind of, I think, good example of, so the top and then the bottom of different types of cells. And you can see the labels at the top and then on the bottom. What do you guys think about, what do y'all think about the difference between these two?
I think one thing that stands out to me in the top row is like someone that's like red-green colorblind wouldn't be able to tell the difference between a cancer and normal cell.
Totally. Yes. That's a really good point. I think we forget often that there's like a good number of people in the population who are red-green colorblind who, for them, the contrast in these figures would be very difficult. So when I make figures, for example, I try not to make them only red and green as like the two contrasting colors, because, you know, if someone's reading, say there's a reviewer reading my paper who's red-green colorblind, they're not going to be very happy about that. They're not going to be able to tell the difference. So that's a really good point. So what if I told you that the idea about this is to show that these cells are kind of related in some way, that there's like some progression? Is there one that you like better than the other?
Jacob, what do you think?
I like, I mean, in my opinion, I like the ones on the bottom, in the bottom row, just because I guess what you were saying earlier about the different colors kind of detracting from the point. I like how it's kind of sort of like a simplistic diagram of like you have the circle and then the different ways to shade the circle. It shows how they're all kind of connected, but it also distinguishes each diagram from the next one.
Yeah.
So I like that.
I think that's like very well put. I think the idea, the thing about the top row that bothers me is that, like, one, I don't know why the immune cell is a hexagon, right? Like, so if you're the reader and if I were reading this, I'm like, why is it a hexagon, right? That's not the point of the figure, right? But it, again, it detracts from the point. Or like, why does cancer stem cell go from pink to red? Like, is that what happens? Right? So it's just makes it more confusing, and sort of leaving things up to the reader or the audience makes them sort of question, like, what am I looking at? So I totally agree with you that in some ways just the shading makes it more simple, right? They're all cells, so they're all circles, and they just have different shadings to denote what they are. So those are other like very stylistic things that I think I often didn't think about until I was like reading some of these papers and like visualization kind of rationale papers that I try to pay more attention to. So color, which we talked a little bit about, is can be used in many different ways, but using color to your advantage is also really important. So this is just the idea that the same color can actually look different depending on the background. So these are actually the same color, and you can see like these two are the same color, but they look different on different backgrounds. And that's just like sort of like a visual trick that your mind plays on you that you should be aware of. And then the other thing is like when you're trying to show different shades of something, depending on surrounding colors can make things look different. So the two rectangles here that have the asterisks, those are actually the same shade of red, but they look different because they're surrounded by different things, right? So that's probably not the best if you're trying to show a continuum of data, that these are supposed to be the same color, but they look really different. I think this one on the bottom looks darker and this one looks lighter, only because this one is surrounded by really dark red, right? That's like very, very subtle, but I think can make a difference in accurately representing your data. There are a lot of other aspects of color, which you can read in like excruciating detail about, which is actually, I think, really interesting. But there's like things called a color picker. You can pick hue, the lightness, color saturation. These are all things that you can play with and just see what it does. You can also see that it's nice to be able to pick colors that are like actually discernible from each other. So I've seen figures where there's like a dark blue and then like a medium blue and a light blue, and you're like, why? They all look so similar, right? So actually having color contrast and then making whatever the most salient point is be the most salient color, right? To make that saliency, relevancy consistent too. But you know, there are also these tools where you can define color groups. So say you're like, I have, in this case, they have 6 different conditions, but you say you have 3, you can actually select color groups that are suitable for the number of conditions that you have based on whether you want a particular color hue or color saturation, or you want to do different colors. There are tools that actually can help you pick those and that are very visually appealing. There's something called Color Brewer that I use a lot for figures that people actually use for making maps, and that are very, very useful for making figures. And then Chloe, you made this really good point about color blindness. So, so if you have normal color vision, you should be able to see the 6. And you know, if you didn't, this is what it would look like on the right, is basically just can't see it. And it turns out that if you can change some of the lightness of the background, that can really improve contrast, where even someone who's colorblind can actually still see the 6 come out of the screen just based on contrast, right? Those are subtle things that make a huge difference. Like what if you had a reviewer who like didn't see the number in the middle and like just doesn't get your point, right? Or a reader that just can't see it. So you also want to make your figures like as accessible as possible. And then these are other things that you can also read more about, but these are different colors with their red, green, blue, numbers. And then there's different types of sort of color blindness indicated here. And this is what sort of simulated colors of what people with these types of color blindness see. So you can see that, you know, if you picked like orange and yellow, it might look really like gross. And so you just lose a lot of the contrast. So kind of being more careful about that. And then some of these go into more detail about like conventional color coding that we use for people with red-green color blindness. So on these natural color images are sort of what you've tried to convey in your figure. And then on the right are kind of simulated colors as are seen by people with red-green color blindness. So this is like an original red and green color coded. And they're just different types of color blindness that they're describing here called protanopia and deuteranopia, but they're both red-green color blind. So that doesn't look very good compared to what you originally wanted it to look like. So if you replace the red with like a magenta color, you actually get a better contrast for someone who's red-green color blind. So that starts to come out as more blue. And then if you replace the green with this turquoise, you also get a better contrast. So trying to think about those things, like I said, I try as much as I can not to do red and green alone, because I think sometimes they can look really strange if someone is colorblind. And then we sort of alluded to, and many of you said this when you were commenting on those figures, was that like this question of do you even need color, right? So we often use color to convey information, or like it's a color that we like, or to convey salience, but I think sometimes we misuse color a lot. And there are these like designers who are very into design aesthetic who say that color used poorly is worse than no color at all. And there's this one guy who always says, above all, do no harm. So be careful, because color can convey the wrong information too. So this is another one of those examples where this scale of color is very odd, because the brightest colors are in the middle, kind of like that other scale that we were looking at. So always ask yourself too, like, do you really need to have color in your figure? And is it really helping to make your point, or is it detracting from it? This is just showing that you can juxtapose colors with different saturations to make certain data appear underrepresented or overrepresented. And so if you look at, for example, the black, the red, and the turquoise compared to the black, the dark gray, and the very light gray, the ones with the very light gray look super, super faint. And it makes it look like very underrepresented because it's hard to find it in the figure. So in some ways, some people might think that's good, but it's sort of exaggerating a feature that might not really be there, right? So it's not perfectly accurately conveying the data as it should be. And then thinking more of like whether you really need color are some of these figures, right? So like, I like putting this one up because it's like so crazy. But I don't know if you guys remember learning about the TC cycle in biology class, which I have nightmares about still. But like, do you really need all these like rainbow colors in this figure? Like, probably not. You know, to the point that I think one of you made earlier, it sort of detracts from the point. The point is just that it's a pathway and it leads into this cycle, right? Maybe there's like one particular compound here that you want to highlight and give color, but whether they really need to be color coded is unclear. So for this example though, the color is related to some correlation value. So they've actually like integrated the color across like multiple figures, right? But if you're going to do that, you need to make it like very, very clear that you're doing that these color correlations like match here. And to me, it's not super intuitive, right? It's not like really telling me that clearly that they match. I just think that someone like went really like nutso on their Illustrator and got really excited about all the colors that were available to them, not that it's related to a correlation, right? So thinking about that too. And then the last few things are about labeling, which is also something that I think people don't really think explicitly about. They just do it without thinking about where things should go. So these are just examples of kind of poor label placement, good label placement, and then label placement priority. So here the labels are kind of everywhere. And the point is to see the dots, right? And that the dots are labeled. But in A, the labels are sort of obscuring that, right? So, and B is an example of where the labels are better placed. So they're spatially kind of like equidistant from each other, and they're all kind of like paired with their dot very closely by a corner, right? You guys all see that? They're all sort of kitty cornered to the dot. Whereas this is like on the side of the dot, on the bottom of the dot, side of the dot, so on top of the dot. So even putting your label in the right place can actually make a huge difference for, like, I know that when I review papers and I see something like this, it just makes me really annoyed. And you don't want your paper reviewer to be mad at you already before they read your paper. And then basically like strategies for removing any guesswork in placing labels. So like where you decide to place labels is also really, really important. And then there's a couple more examples of labels here. So this is basically like avoiding an A encoding the same information twice. So there is species that don't need to be made by both color and by this code. So it gets kind of confusing, right? Because it's like FU-CLDN 25, FU-CLDN 10E, like why are these both green, but they're like not related, but they're, you know, so this is basically too much coded information. There's too many labels. So either you should just do the text or you should just do the color. It just makes it a little bit too confusing to follow. This is like a different example where the labels are much shorter. And so people are able to parse through the information faster, right? M15 is faster than reading through all of that. That may be easier. And then you can kind of like integrate some of these design principles, right? So what if you actually line these all up so that they're each aligned? It's like even easier for me to read and faster for me to read, right? So, you know, these are all pretty similar, but I do think that these two, B and C, look a lot better. And they're easier for someone to read if they're just looking at your presentation or reading a paper or your proposal. And then the last thing I'll say about that I'll talk about is just these labels. Do you guys have like a sense of kind of the difference between these two?
I can explain the difference, but I'm not really sure which one is clearer.
Yeah.
It looks like most of the lines in B are horizontal, except for the IIC cells, whereas the lines are sort of multiple different orientations in the top figure.
Yep. Spot on. Anybody else have comments about these two? It's very subtle difference.
The cells themselves are arranged differently also in both of them, but I'm also not really sure which one is more effective.
I think there are arguments to be made about either one being effective. I think I tend to lean a little bit more towards B. So you can see that it has that grid structure, and the grid structure is kind of put in place for you to see what the grid is, the person who made it, so you can actually like see where the grid was placed, and this curvilinear grid, but everything's just much more aligned. And so it just becomes easier to see, even though it's very, very, very subtle. So some of these like figure changes are really, really minor, but do look a little bit better and just a little more simpler, right? So here the parasite has this like angled line, but here it's straightforward. These are not aligned. Here they're all aligned, right? These are definitely not aligned on the curve, and this one they've actually all been aligned onto the curve of this, like whatever this structure is. I guess it's just a group of cells, right? So, you know, and then for some reason this one, immune inflammatory cells, is on like 3 different lines. So these are like really, really subtle things that even like very seasoned scientists and figure makers often don't think about. But I like to show this one last because I think it's, you know, they're barely different, but they are different. And the bottom one just incorporates a lot more of these like more strict design principles and visualization techniques that I would encourage you all just to think about when you're making figures. So I think I'll stop there. I would highly recommend checking out these papers. There's like many, many more on like, there's a whole paper on like axes and tick marks on your graphs when you make figures. Like these are like the minor details that make a huge difference. And I think just being more conscious of them can make your data look a lot better.
Awesome. Jessica, it's always refreshing to focus on the fundamentals. So for those of you who are watching that, you notice how fundamental it was, right? Some things you brush over, you want to hear it once. No, you want to hear this as often as you can. And practicing those elements over time makes the difference, right? That reviewer is able to understand what you're trying to say. That reader is able to get, that audience member who's looking at your presentation is able to understand. I'm always a firm believer of less is more. And figures that are trying to just clear one thing, you know, I think I showed you all the citation or number of publications on SciComm, the figure a couple of weeks ago, just one direction, right? It's like one message, that was it. So hopefully that was useful, okay? You guys are gonna have diverse data sets or different types of data. So it's gonna be up to you how you wanna communicate that. All right, and in the next couple of weeks, I'm curious to see how the different figures come into play. You may have one figure, that's fine, right? Or no figure, because your data set is like different in some other way. But just keep in mind, ask yourself, what is it that I'm trying to communicate, right? I think that's what Jessica's been talking all along. What are you trying to communicate? And keep it simple, right? The elephant in the room, just point at it, all right? Like it's in the middle of the room. With that, Jess, thank you so much. Any questions, any final questions for Jess? She's of course on Slack. You can message her anytime if any questions.
I just put the link into the Nature blog post. It has all of the data visualization papers. So I like looking at them. They're just fun to think about and look at. So
yeah, and just in terms of tools, Illustrator, right? You use Illustrator?
I use Illustrator for all my figures. Yep.
Okay, anything else you use like?
If you become more advanced, you can use like programming languages to make figures. So when I have like large volumes of data, I use like something called ggplot on R Studio that makes very, very like aesthetically pleasing figures where you can basically tailor make every single like thing that you want. Those are much more complex, but Illustrator is like my go-to for most things.
Prism, of course, is another one for its quick data plotting and so forth. Raw graphs is another cool figure. So all these things, by the way, guess where they are? Guess where they are? In our library. So again, I think all my slides so far included a visit to the library. I don't have it today, but please visit the library because we have a collection there of tools, and ggplot is there. There is also some other, R graph I'm seeing here. Jess has put it together.
Yeah, the color brewer. So if you want palette inspiration, I like that.
Yeah, and the cool thing is to keep practicing experimenting. You'll get some insight. You get data set, just mess around with it until you see the pattern, the key message you're trying to communicate. We did this last year with the intern Riley. We went through several iterations. I mean, several iterations. And I want to just show you this quickly here before we adjourn, because it ended up being quite an exercise of practicing. And goodness, I don't know where disappear to, but give me one moment. Because I think all the principles that Jess was talking about, we were able to try to put into practice. Oh, here it is. So this was, again, she had data sets, like big volume of data she was playing around. But just think about what Jess has been talking about, the alignment, the colors, like just trying to figure out what is clear, what is, making it very simple. And here, the same data here is just plotted two different ways. Okay, same data plotted different ways. And so you are able to show the viewer very different points of view and different ways to look at the information. So I'll spare you the rest with that. Have a good week. So next week, we are going to do office hours instead. They'll be optional. Feel free to stop by here, 4 o'clock. I'll be around if you want to stop by, but we won't have a formal lecture, okay, or a workshop. The following Saturday, that's part of the reason, because the following Saturday, we're gonna have our symposium. So I want you guys to save your brain power for that, okay? That's gonna be a lot of fun, a lot of speakers, and you're gonna get a sense of the full breadth of, quite a wide breadth of people's projects and what they're working on, and the questions they're asking in their practice and also in their research. And of course, Bruce is an awesome, will give us a big landscape analysis of where have we been? Where are we? Where are we going? So I'm really curious to get that perspective from him. So again, remember next week, office hours Tuesdays, okay? Have a great 4th. Relax, keep cool wherever you are. Keep cool. I think we're all cooking. And of course, find us on Slack. Feel free to ping me at any time. Jess, thank you.